some of these electron carriers outside, and this is in the mitochondria already, okay? So the electrons need to be passed on. So how do we do? How do we pass on, okay? The pass on. So we do this by using membrane-bound protein complexes, and these are electron complexes on the inner membrane, yeah? And we have in extra 2 mobile electron carriers. Okay, it's easier if I describe it with this picture over here. Okay, so you have the outer membrane, freely permeable to small molecules, ions, etc. We have the intermembrane space, and then we have the inner membrane, which is not permeable to most things. Okay, so now these lumps here are proteins, and we call them protein complexes. Complex 1, complex 2, 3, 4. These are the electron transport complex. Right? So they do the job by undergoing subsequent, consecutive oxidation reduction processes to pass your electrons. Okay, the concept, yeah? So NADH will be produced in the matrix, right? Because of citric acid cycle, and it will pass on its electrons, right, to complex 1. And once complex 1 receives electrons, it will be reduced. To regenerate complex 1, it has to be oxidized again by complex 2. And then, by Q, which is ubiquinone here. Right? So over here, electron will be passed directly to this mobile one. These are fixed. 1, 2, 3, 4 are immobilized on the inner membrane. Q and cytochrome C, right? Q, which is coenzyme Q, and cytochrome C, they are mobile. They are the floating ones. Okay? They are the floating ones that can move around. So by doing this, the electrons get passed to Q. Q passes it to 3. 3 will pass it to cytochrome C. Again, okay, they do it by oxidation reduction. Okay, 3 will get reduced to receive the electron, and then it gets oxidized again by cytochrome C. And then finally to 4, which will then pass it to oxygen inside the matrix to give you water. Right? Oxygen is not an issue, right? Oxygen permeability is gas. It's a gas, so it can go pass through anything. Right? Gas is permeable to gas. So oxygen is here, and the electrons will be passed to gas, it becomes water. This is the final step. Okay? So the special thing about complex 2 over here, remember just now I said that succinate dehydrogenase uses FAD instead of NAD. And succinate dehydrogenase in this case is mobilized in the membrane together with complex 2. So do you see this reaction? One of the TCA reactions is over here, succinate to fumarate. This reaction occurs very near the membrane. Okay, all the other reactions is here in the matrix. Okay, in the matrix. And over here, this particular reaction occurs have to be close to the membrane because the enzyme is mobilized here together with complex 2. Right? So once FAD, which is here, gets reduced to become FADH2, the enzymes will be passed, the electrons will be passed to Q, coenzyme Q, and follow the same procedure to give you water at the end of it. Okay? So what does this do for us? It's just passing on electrons and giving us oxygen. Is that it? That's not it, because throughout the whole process, if you notice, NAD, and this is not written anywhere, but if you look at the reaction, together with the oxidation reduction process of NADH, electrons is involved, yes, there's a pair of electrons. It doesn't take up one electron, right? But protons are also involved. We are forgetting the protons. While we are passing the electrons, where do the protons go? Right? Where do the protons go? There will be an accumulation of protons here if you don't remove it. And what happens? When you have accumulation of proton, what happens? pH changes. And you know pH has a very tight, a lot of processes or solution reactions are affected by pH. So the body must have a way to pump out the proton, okay, to maintain this homeostasis, to maintain the environment. So while electrons are being passed down, protons are being pumped out, okay, because we said that this membrane is so tight, even protons cannot diffuse freely, so it needs to be actively pumped out into the space, right? So for every mole of oxygen or molecule of oxygen reduced, 10 protons are being pumped out, okay? And this kind of pumping out generate what we call proton motive force. Why? Not just the proton, not just pH changes, right? Proton is always associated with pH. Wherever the protons are, the pH will reduce. It is also associated with electrical charges, okay? Because it's positively charged, right? So not just chemical energy, but you also have electrical energy being established across this membrane, okay? So I added one more slide like this. Okay, this is what I mean. So you have the inner membrane, you have the proton pump. So this is the matrix, N is matrix, pumped out into the intermembrane space. Proton will move from here to here. You see the accumulation of protons, and then you have OH minus here. So this will give you a pH change, but also charge changes. This will be all positively charged to be negatively charged, right? So this movement of protons will give rise to a chemical potential and also an electrical potential, right? And the net effect of this potential is called the proton motive force, which could be quantified, okay, by this term called delta G. It's the same delta G that we have seen. You don't need to memorize this, we're not going through this. But the idea is that the delta G is here. Okay, so let us go back here. So now we know that there's going to be a force, there's going to be tension, right? A lot of tension on this inner membrane. Protons go out, and then there's electrical potential. Now, if it doesn't come back in again, we have a problem, right? If it doesn't come back in again, this reaction will potentially stop. Why? Because the energy that is being built up here is so strong that it cannot be overcome by the transfer of electrons anymore, right? So this whole process will stop. That it's like an energy barrier, right? You try to move the electrons by pushing them through, and yet there is a huge build-up, okay? Huge build-up of protons here, okay? It's a huge energy barrier that you have to go through. It's like an activation energy. So once it is too high, this cannot happen. Oh, then what happens to mitochondria? It dies, okay? Basically, it is the end of the process. You will not continue, right? And it's the end of energy production. So in order to stop this from happening, you must have a way for the protons to come back in again, okay? Pump out, and there must be a way for it to pump back in again, right? And this process is then facilitated by what we call an ATPase, F0, F1 ATPase. Again, it's another channel. It's a pore, it's a transporter. You see the hole here, okay? This transporter serves specifically to move the protons back in again to relieve the buildup, right? To maintain the pH across the membrane, okay? And it has been found, because of this movement of protons back in, this energy is then conserved, right? We don't waste the electrical energy. This energy is then conserved by using it to make ATP hydrolysis, right? Remember we said this is about plus 30.5 kJ per mole to happen, right? So it's just nice that this electrical energy can power this ATP production. Okay, so whatever that is written here is what I have described, right? The protons are being pumped across the inner membrane, and the gradient, difference in the proton across the membrane gives you a voltage gradient, and this gradient is coupled together with oxidative phosphorylation. So what I have described to you is the whole concept of electron transport, where I'm not going to rush through oxidative phosphorylation. It's quite a lot to understand. So I'll just stop here today. Okay, so I've covered Krebs cycle and then the electron transport. Don't leave yet. I think Jun Hao wants to make an announcement.